Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Joseph Mazzulli, Associate Professor of Neurology at Northwestern University. Joe is also a 2022 Toffler Scholar. Today, we're going to discuss Joe's research in determining how protein misfolding and amyloid formation results in cell death. So let's dive in. Welcome, Joe.
Thanks for having me.
I'm excited to get caught up. It's been a while since we chatted about your research, and so I'm really excited to hear what you've been doing. I would like to just kind of jump right into your research laboratory and its focus, and really, what is protein misfolding and amyloid formation? And I know you're working with special stem cells to research this, but can you explain what the focus is?
Yeah, sure. So our lab is focused on the main question is how proteins convert into this normal folded state into a misfolded state. And that often will lead to the formation of an aggregate, which in the case of the neurodegenerative diseases that we study, like Parkinson's and Alzheimer's disease, it forms this particular structure called amyloid. And that's just a term that indicates that the protein is insoluble. It's aggregated in a specific conformation, which is rich in beta-pleated sheet. And it somehow, when it aggregates, it results in neuronal cell death. And that's what we are studying, those mechanisms of how the aggregation process is triggered, the initiating factors, and also downstream of that, once those aggregates are formed, what essential cellular processes does it interrupt? And we use mainly induced pluripotent stem cell, iPSC models to study these questions.
So I've had other people on and we've talked about stem cells. What's the difference in this, and I think you called it pluripotent stem cell? What is that? And, how is it different than maybe what other people know as stem cells?
Induced pluripotent stem cells are made programming process that was discovered 15, 20 years ago. And they're generated through somatic cells. And often the source will be skin cells, for example, a skin punch that you can get at the dermatologist, or blood cells as well. There's white blood cell fraction that we can get that we can reprogram. And what it's basically doing is sort of winding back the clock. You're starting with a differentiated cell. You're adding in a cocktail of factors that revert it back into a pluripotent state, which is what it started as. And that's an advantage because once we induce those cells into a stem cell, we can differentiate them into any cell type that we want. And the main cell type that we use are neurons. Neuroscientists will often do glial cells, astrocytes, microglia, for example. But what we study in the lab are differentiating these iPSCs into neurons.
So so what I heard you saying is then the stem cells come from the individual, and then you reprogram them back to what they originally were before they became whatever they became when you took them, right? And that you're trying to, and you're doing that because that it gets more precise because that's the individual versus more generic stem cells from other people and trying to understand the disease. Am I understanding that correctly?
Yeah, that's the major advantage. And I think for for disease modeling, which is what we mainly use these for. They could be used for multiple purposes, regenerative purposes, for example, and disease modeling in the lab, which is what we do. So you're taking the cells directly from a patient with a condition like Alzheimer's disease or Parkinson's disease or other rare genetic diseases. And that's important because you have all of that patient's genetic information in the cell, and you could study how, for example, in genetic diseases, you can study how a pathogenic variant leads to cell death in culture, and allows us to examine mechanistically the biochemical processes and the cellular processes that go wrong as a consequence of that pathogenic variant. The other major advantage is that once you reprogram those somatic cells into iPSCs, it basically gives you an endless supply of those cultures that will last forever. That's important for us because there are some techniques and assays that we use that require large amounts of material. It's hard to get some of that stuff, primary cells, for example. If you wanted to study skin cells from a patient, you could really only get so many of them, and then they end up dying off in culture. These are immortalized, so they will last forever. We freeze down stocks in liquid nitrogen that will last indefinitely. And as I said, it gives us that opportunity to scale up the amount of material that allows us to do a lot of different assays and analyses that we otherwise would not be able to do with primary cells. And besides that, for neuroscience, there's no way we're going to get brain material from a living patient. That's very difficult to do. And so this gives us an opportunity to sort of model what certain cell types or certain neurons are doing in vitro.
Yeah, that's really interesting. And I'm sure we'll talk a little bit more about your work in trying to understand possible therapeutics and kind of testing on that. And I would imagine that becomes very important too. I want to go back to amyloid and this misfolding of this protein. There's a lot written about amyloid and tau, and it, from the layperson, it would seem like doctors and researchers understand this already, that there's common knowledge about, these proteins. Why are we still studying them? And, because I think that's an important question for other people that maybe, not understand, is like, why are we still studying this? Don't we understand?
Yeah, there's a lot that we do understand. A lot of that comes from rare familial forms of these diseases like Alzheimer's and Parkinson's disease. But most of the cases there are sporadic. We have no idea genetically what is initiating the disease. We do understand that pathologically proteins in this amyloid-like conformation accumulate in the brain. In many cases, we don't understand why. What was the trigger that started that? There's a theory that it's a combination of genetics, complex genetic risk factors that a patient is harboring, possibly combined with something in the environment. And so that interaction, if it's just right, can lead into protein aggregation and accumulation. So that's really the big question. I think that we still don't really understand. How do you get from a normal soluble conformation into an insoluble conformation? So as I mentioned, there are some very rare familial forms of Parkinson's disease, and we understand how protein aggregation occurs. We understand more, I would say, in what's causing the aggregation in those diseases, because there's specific point mutations that we know in certain proteins. In Parkinson's, the main protein is alpha-synuclein, and that's the main protein that aggregates, and many people have been focused on because it's a major component of the inclusions that you see in the patient's brain when they die. So in the genetic cases of Parkinson's, in some of them, there are point mutations in the alpha-synuclein protein that actually trigger its conformational change. So that's the part that we do understand. But as I said, in sporadic disease, we don't understand how the aggregates are forming. Why do they stick around in the cell? Why are they filling? They essentially fill up neurons in their extensions and in their cell bodies and form a solid. The cell should be fluid. This is more of like a solid material that it's essentially filling up the neuron, and we don't understand how that's happening. And downstream of that is also, I think, a big mystery. And so the idea there is if we could stop it or find a way to clear out those aggregates, those are the mechanisms that we study in the lab, how to get rid of that aggregated material. Then that will eventually lead to some kind of disease modifying therapy.
That's interesting. And I appreciate you going through there. I think there's a lot of information for people to sift through and try to understand. And I know for researchers, there's a lot too. But I think that question of why is it happening is the big search. So how did you get to where you are? Can you tell us a little bit about your journey and how did you become a researcher and why this focus?
I started studying neuroscience as an undergrad in probably 1996 or so, and I came into the University of Pittsburgh as an undergrad, majoring in biochemistry, and then as I was taking electives, I discovered neuroscience, and I became hooked after an intro to neuroscience class. I guess, what intrigued me about that was that the class really sort of presented these brain mysteries, patients with odd behaviors, and then going on to explain neural correlates for those behaviors, problems in specific parts of the brain. Sometimes it's from an incidental injury or a car accident or some type of brain injury or a procedure that went wrong, and explaining how these behaviors occur at the cellular and molecular level as well. And so that's part of why I wanted to be a part of that, to help figure out these brain mysteries. When I became interested in neurodegeneration, was basically stemming from interest in protein chemistry. I was always interested in how proteins maintain solubility and what triggers their conversion into these insoluble proteins, the conformational change that occurs. And of course, a lot of the neurodegenerative diseases, protein aggregates are a major histopathological hallmark. And even in normal aging, there's proteins that accumulate in the brain. And that's sort of how I got interested in that topic, stemming from basic neuroscience and then sort of fusing it with protein chemistry. And then I just thought that it would be really an interesting project to sort of connect the dots there on how protein aggregates form and how they then cause a neuron to degenerate.
Do you have, because I think these diseases affect most people, their families and people that are there people that are within your family circle that suffer from these diseases?
Yeah, mostly we've been, in our immediate family, we've been pretty clear. I had a grandmother that passed away a few years ago from Alzheimer's disease, and she lived to be 95 or 96. But she was always very sharp. And then all of a sudden, things just sort of went downhill. And when you experience it yourself, it happens so quickly that it's such an unbelievable experience when your grandmother doesn't recognize you and no longer knows who you are. But Alzheimer's or Parkinson's, I think, combined, it's so common over a certain age that I think that there's, almost everybody you talk to will be affected by these diseases.
Yeah, you mentioned, that these are age related and, kind of the normal aging process. I've asked others, is it a given that these diseases will, manifest as we get older or because of, just aging, or is it there's something that we're doing as we're aging that is leading to these diseases? And that's kind of a different perspective, I think, on the disease. What, as somebody that studies this, what are your thoughts on, because you mentioned the environmental, and there's environmental inside our bodies or environmental in what we're, how we're living. What are your
what we're
thoughts on
exposed
that?
to.
Yeah.
That's a really great question, because there's a small percentage of people that survive to 90 and over 100 and are mentally very capable, super agers, they're called, right? But most people, I think, experience cognitive decline as a normal part of aging, movement balance problems and slowness of movement as you get older. I do think that most of the population, if they did survive into 80s, 90s, and even to 100, that you would see these diseases a lot. If they didn't die of other things like cardiovascular disease or cancer, you would see these diseases coming up a lot more, because there is something that happens in the aging process that we don't understand. As we age, we lose this ability to maintain homeostasis in the cell. What we're most interested in is protein homeostasis. This is the balance of how proteins are synthesized and degraded. And something along the way perturbs this process, and we think the degradation part of it is probably what's causing problems as we age, in the sense that the cell can't degrade the proteins fast enough. They then accumulate. And then certain proteins have this aggregation prone property. It's just something that's intrinsic to them. They could be very abundant naturally in our cell. They could have amino acid residues that cause them to stick together, hydrophobic residues that cause problems. That's what we think the case is for alpha synuclein that accumulates in Parkinson's disease. So over time, we don't understand why, but that ability to degrade proteins is compromised. And sometimes there's inclusions that we can tolerate in our brain. There's a certain level of damage that you can accumulate, and you will not see any clinical phenotype. It's interesting to think about Parkinson's disease, where midbrain dopamine cells degenerate. And it's thought that, people can tolerate 60 to 70% of the loss of those cells before you start to see any symptoms at all. So thinking about that, and that cell loss of dopamine, that occurs naturally over age as well, which is what leads to the slowness of movement and imbalance problems. But I think it's a really interesting question. It definitely could, we think, have something to do with what you're exposed to, head injuries and things like that. Pesticide exposure has been something that we've studied in the Parkinson's field. Certain things that may increase the oxidation or inflammatory cascades in the brain. And so all of those things, I think, depending on what you're exposed to and what your genetic background is, we think that there are susceptibility factors that will cause some people to have Parkinson's disease while others can live into their 80s and 90s and not exhibit any clinical phenotype at all. And something that we study, and we can talk about more in detail later, is the cellular degradation, the lysosomal system. There's many gene variants in that system that we think are causing an increased risk for developing Parkinson's disease.
Yeah. So why don't we just go into that right now on that variation? Because I was actually going to ask you, because you're talking about, like these very rare diseases, neurological diseases. You're talking about, kind of the portfolio of Parkinson's, if you would, the different kind of Parkinson's, and then Alzheimer's. You're studying the suite, I guess, if you can call it that, of these diseases from the cellular level. And, Do the different kind of breakdown of the cellular function, does it lead towards a different disease, or is it, is it one of those random things that as it's breaking down, we don't know which disease it's going to point to, or is it pretty known, how that works in the cell and what kind of disease it will manifest?
Yeah, in some cases it's known, and in some it's not known. It could have to do with combination of several different risk factors that a person may harbor that will dictate whether you get one disease or another. The important thing to know, though, is that a lot of times, many times, you cannot get a definitive diagnosis of dementia or Parkinson's or Lewy body dementia until you're at until a pathologist examines the brain post mortem. Then what they do is they look. The pathology is very important in diagnostic procedures, along with the clinical phenotype. If they see a certain pathology and accumulation of Lewy bodies and alpha-synuclein aggregates, and it's in certain regions of the brain and not in others. If it's in midbrain dopamine neurons, but nowhere else, then it will help in defining what you get. But in terms of how the genetics is connected to those conditions, I think there are certainly genetic risk factors that are associated with one disease and not the other. We study a lysosomal gene called GBA1 that is a very strong risk factor for developing Parkinson's disease and Lewy body dementia, and as far as I know, is not associated with Alzheimer's disease. But the comorbidity and the mixing between those two is something that is a topic of research in a lot of labs, and we're starting to study that as well.
So what is the kind of the variance of what you're studying and how you're studying? You mentioned these variants. Can you talk more about that?
The variants that we are studying we really focus on only a few. The lysosomal GBA1 gene is the main variants in the GBA1 gene are what we study in the lab. So GBA1 encodes a lysosomal enzyme called glucocerebrosidase, and it degrades sphingolipids in the lysosome. What we know about and homozygote carriers or mutations in this gene cause a rare disease called Gaucher's disease. And so what we know is that once the sphingolipid substrate builds up, which is due to a loss of function in the enzyme, that it somehow causes cell death and leads to neurodegeneration. So that's one of the major risk factors for Parkinson's disease. These are patients that only carry one single copy; they're heterozygote carriers of the GBA1 gene. So they're not going to develop Gaucher's disease because they have one functional copy and they have one dysfunctional copy. But what genetics has told us is that they are at increased risk for developing Parkinson's as they get older, and usually it's earlier onset compared to a non-GBA1 carrier. For example, age 40, 50 may be the onset, and they're also more likely to have a dementia phenotype. That the disease is more likely to go beyond the movement system, the midbrain, and affect the cortex and cognitive functions more dramatically if you have a GBA1 mutation. That's the main variant that we study. There are a few other lysosomal variants that we have been looking at, and we also study rare variants in the proteins that in the alpha-synuclein protein that is the main component of Lewy bodies.
Yeah, so Lewy body, when you say Lewy bodies, this is leading to the Lewy body dementia. And what can you, or where does that go? Like, how are you studying and what are you studying it for?
So the Lewy body is a term for the pathological inclusion that characterizes both Parkinson's disease and also Lewy body dementia. And there's a few other neurodegenerative diseases that you see Lewy bodies in. So the main distinction between the two is how soon you get the dementia phenotype. But there's a lot of overlap there between Parkinson's and Lewy body dementia. We think that the common feature is that certain neuronal subtypes are somehow susceptible to an overabundance of alpha-synuclein, which allow it to accumulate into these aggregated forms. It does form an amyloid-like structure, which is what you see as a part of the Lewy body. We haven't been able to directly link Lewy bodies with toxicity there in the brain. So that's the thing that distinguishes them. You can't find Lewy bodies in healthy controls as well, healthy patients, but it's not as abundant, of course, as it would be in disease. So, in the lab, what we're looking at is sort of the common question of how some of these genetic variants lead into the aggregates of alpha-synuclein. We can't form precise Lewy bodies in vitro in our iPSC models, but they have similar features. Biochemical solubility is similar. They do have amyloid-like properties. There's certain dyes that we can use that we can tell if they're in the amyloid-like state, and they're building up in the cell. And we have different ways of tweaking the cultures to see what would make it worse and what makes it better. And that's essentially how we look at mechanistically, how the disease process is starting.
So Lewy bodies are things that are naturally occurring. We need them for some function, but they're actually building up and creating that damage to the cells, and we just don't know why that is. Did I understand that correctly?
I wouldn't say that they're needed for a function. There are certainly some functional amyloid proteins out there, but I don't think synuclein, as far as we know now, is not one of them. This is a process that happens pathologically. I mentioned before that you can find Lewy bodies in the brains of aged individuals that seem to be healthy, no neurological phenotypes. So I think there's a certain level of injury that our nervous system can handle. So that's not to say that it's a normal process or that we need them. But for some reason, they do require age and likely a combination of genetic variants that causes them to build up and induce neurodegeneration in the nervous system.
And you're studying the genetics, you're going back into the genetics to try to understand what those common factors are for some of these diseases.
Yeah, we do study some genetic variants, but a lot of the mechanisms that we look at are focused around how lysosomes degrade proteins. So they're all a few of those variants that I mentioned before, like GBA1. How when their function is lost, how does that induce protein aggregation and cell death? But our lab mainly, we're not in the genetics discovery field, so we actually rely on geneticists quite a bit to look at patients and find new discoveries in different proteins that are associated with disease, and then try to model some of those variants in our cultures.
So let's talk about your lab a little bit more in detail then, and kind of the network, if you would, of collaboration. You're working with geneticists. Are you working with clinicians? How does that work? And I'm especially intrigued with this, with Lewy body, because there seems to be misdiagnosis, if you would. Is this Parkinson's? What is this? And because it's just kind of a new area that, at least from what I understand, is, the last 5 or 6 years, more emphasis being put on trying to understand what is Parkinson's, what is Lewy body dementia. What is that network that you collaborate with to get your work done?
That's a great question. I think we do rely on geneticists, as I mentioned before, for discovery of pathways and variants that could lead into faulty pathways in Parkinson's cells. For working with clinicians, there it becomes more when a lot of our studies are basic science focused, which means we're starting at the molecular level and we're trying to determine if a protein is involved in protein aggregation or disease process, lysosomal dysfunction, some downstream phenotype that we look at. When we find a therapeutic target and we have some way that we think we can drug it with a small molecule, that's when we sort of go to the next phase. We start in cell culture. We use patient-derived cultures. We have specific phenotypes that we perform in the lab that can tell us if there's a beneficial effect in boosting the activity of our putative target. And then we often go to animal studies to see if it's a small molecule, for example. We ask basic questions. Does it get into the brain? And if it does, is it engaging its target? And again, we're looking at do protein aggregates go away? So that's our ultimate goal. If we reach that stage, then we reach out to clinicians and discuss the data, all the preclinical data with them, and determine if it's feasible to move on to a phase 1 clinical trial. Some of the compounds that we're using in the lab are FDA approved for other indications, like blood pressure, for example. Or some of them have gone through some clinical testing for other diseases like cancer and have just failed for cancer. But we know that, they're safe for people to take. They didn't have an effect in whatever disease they were tested in before, but we know that at least they're safe. So that actually helps a lot because that cuts a lot off the time and the cost. So there are some small molecules like that we are studying that are sort of ready to go for the clinic. But that's where we connect with clinicians, and I think when it comes to readouts, one of the important things that I think we need to maybe improve upon a little bit in the future are biomarkers, indications that things that we can measure from accessible fluids like blood that tell us that we're sort of on the right track or we're not at all. And that's, again, we don't do biomarker studies in our lab. We're all basic biochemistry and cell culture. And so that's another opportunity there to collaborate with clinical scientists.
Do people ever contact you to volunteer their tissue? If they've been diagnosed with something, do they ever contact you to ask for you to test it and to try to understand and apply some of these therapeutics to just see what's going to happen?
Yeah, that happens not that often, but occasionally we will have patients with, most often it's rare diseases that sometimes they're diagnosed, sometimes they're not diagnosed, and offering their cells. The connection would be most often if there's a suspected problem in protein clearance or the lysosomal system, or if there is some connection to protein accumulation, because we have the assays that are set up to look at that. And so what we would do is we would collect the patient's fibroblasts or blood, reprogram them in the lab into induced pluripotent stem cells, and then we would convert them most often into neurons, cortical neurons or midbrain dopamine neurons, depending on what the what the disease is. The first thing we do is try to identify a phenotype. Is there some change from a normal healthy control? If we know what the genetic putative mutation is, the pathogenic variant, then we'll try to correct that. We'll look at that gene or protein directly and just measure it. Is it there in abundance or is it gone? Is it loss of function or is it a toxic gain in function? So answering these first basic questions of mechanism. Sometimes it's already known because there's other cases of pathogenic variants in a similar gene where you have something to go off of, but sometimes it's not known. So you're starting from square one. But once we identify phenotype, the next goal, of course, is to try to correct that phenotype in some way. And often we start with trying to overexpress a protein that we think is lost. We try to replace it with a functional version. If there's a mutation in a certain protein, we'll express the wild type version of it and see if the phenotype goes away. And then if we're lucky, the target that we're looking at, it doesn't have to be direct always either. Sometimes we have indirect ways of enhancing the lysosomal clearance system. So depending on where the problem is in terms of the sequence of events in the cell, going from production of a protein to its trafficking, to its initial working in its final destination. So depending on where the problem is in that sequence of events, we have ways of potentially fixing that. But that's been something that I think has been really the opportunity to get cells from a patient and possibly develop a personalized or customized therapy for that person is something that I think is really exciting. And part of what, for me at least, induced pluripotent stem cell, the discovery of generating that cell type, that's a big part of that is to this opportunity to develop personalized therapies for people. We can take a person's cells and find out if they respond to a potential therapy or not. And in that way, we can say, yes, this is working for you, or no, go on to something else, or we can continue to test other options. But that part of it is what I think is the most exciting part of iPSC modeling, this potential for personalized therapies.
Some of these diseases are a race against time. How much time, if people were to do that, how much time does that, I mean, in general, because it's all, it's, you can't be real precise there, but, like how many therapies would you start to test? Like, is that infinite? Is that, are there a handful, 20, 30 that you just start with and then how long does that take? I mean, give us some idea of how does that look in the lab? It just, because it's hard to kind of grasp how much time and what would be going on if you were to kind of execute that, if you would.
Yeah, it's a good question. I think that a lot of times there's administrative things that we have to overcome in order to get material and material transfer agreements and things like that, really slow things down. But if, let's say, all of that works very quickly, from the time we get cells in the lab, we're able to generate induced pluripotent stem cells from a patient's somatic cells within a few months. And from there, we will differentiate the cells into neurons most often, and that takes probably another 2 to 3 months in culture. And depending on how big the problem is or how mysterious the disease is, it could take a while to identify a phenotype in vitro. But if it's something that we're used to looking at, substrates accumulating like they do in lysosomal diseases or proteins building up, we may be able to identify a phenotype within 6 months. So it's possible that if everything works right, within a year, we'll be able to get moving. And some things can actually be done without iPSCs. You can identify, for example, lysosomal storage diseases. You can look at storage in skin, and so all you have to do there is culture the primary cells and find ways of fixing the problem in that cell type, and that makes things a lot faster. But for neurological diseases, we often want to go to the neuron because neurons are sensitive in the sense that they don't divide like other cell types. When a cell divides, it has that opportunity to dilute out junk and storage material. So often, cells that are dividing aren't as affected by protein accumulation diseases, but neurons don't divide. They're postmitotic, and they therefore rely on the lysosomal system very heavily to get rid of their junk because they cannot do it through division. So that's why we often go to neurons. Neurons grow slow. They're more difficult to work with than other cell types. That's all those things slow down the progress. Once we have identified a phenotype and fixed it, then of course it depends on what that therapy is. If it's a small molecule that's FDA approved already for some other indication, then it can be translated into therapy or tried out at least really quickly. If it's another type of treatment like an antisense oligonucleotide, for example, which are little short fragments of nucleic acid that essentially can patch up genetic mutations and correct, improve formation of the wild type normal form of the protein. Some of those are used in other diseases, and so if they've already gone through FDA approval for those diseases, and it makes it a lot faster to translate it.
That's fascinating. And I appreciate you going through there because it gave, a little bit more definition of how hard this is. It isn't just, hey, donate some skin and we'll put it in a petri dish and, we'll start adding things to it to test. It's quite extensive. And I'm sure there's a lot of pretty unique technology too that you have to use to do this. Is that correct? And what are the advancements that are happening in technology that are going to allow you to maybe even accelerate some of your research?
Yeah, I would say there are more things that allow us to do assays in a high throughput manner that have really been developed in the last couple of decades. That's accelerated ways of different treatments that we can do, looking at different conditions, different cell types, and incubation times, and things like that, that really facilitates things for us. So that's part of, I think, at least for us, a lot of the assays that we do are pretty simple, but that's the high throughput technologies I think have helped us a lot. We actually get a lot done through extracting cell cultures in different buffers with different detergents and doing basic biochemical assays like Western blots. We go a long way with that, which isn't what I would say technologically advanced, but it gets the job done. It's still a clear way of figuring out if something wrong is going on. I'd say the other nice thing, of course, is genetics. We're able now to do whole genome sequencing and sequencing of RNA, the transcriptome, a lot faster than we ever have before. And it's a lot cheaper as well. We're getting better at handling these large data sets and analyzing the genome and the expression patterns in cells through RNA and also through proteomics, many types of omics studies that we've been recently doing. It really gives you this global picture of what is happening in the cell. It allows discovery-based science to be done. And a lot of times you have a hypothesis, is one particular protein changing or is one pathway changing? When you do these omics studies, it really allows you to just say, well, let's see what's different and let the cell tell us what is wrong. And that's the advantage of the whole genome sequencing and RNA-seq and proteomics. And I think for us, that's what we've been most excited about.
Does artificial intelligence or machine learning or any of this kind of technology that we all hear about in the news, does it help you with that big data and those data sets? Does it help accelerate some of the discovery of what the cell's telling us?
Yeah, and a lot of labs are starting to use AI and machine learning to identify pathways and variants from genetics and RNA-seq studies. We haven't really done that yet. We haven't gone into that yet. But it's something that I think, we're going to be facing in the future for sure as that technology advances, and many labs are doing it now. We have just really begun our lab has just really started to look at these big data sets, the omics. And so we haven't gotten into the artificial intelligence yet, but I'm sure we will.
So you talked about kind of personalized therapeutics, and that was something that you were kind of hopeful for. When you look out 5 or 10 years, what do you see, and what are you the most excited about?
Yeah, I think we have a few pretty good hits in terms of therapeutic targets and pathways that we're trying to build upon. Preclinical data using patient cultures, using some mouse studies to determine if these therapies and small molecules are engaging lysosomes in the brain and degrading proteins. Are they able to amplify the lysosomal system? But I think, if we're looking 5 to 10 years in the future, if we can get one or two of these molecules into a clinical trial, I think that would be a good goal, a good milestone for us. But the other part of it is mainly a lot of our research is almost all of it is very early stage and basic science focused. So we're really hoping to get more insight into how protein aggregates accumulate in neurons. What we think is that once an aggregate is formed, it behaves in sort of like a virus. It has these interesting self-propagating features. In one way, what it does is if you have a misfolded protein sitting in a neuron, it can interact with a normal form of that protein and convert it into the pathogenic form. And that's in one way that it can seed and propagate a misfolded conformation. The second way that we're finding out now is that these protein aggregates can actually disable the cellular process that is meant to degrade them. That's the lysosomal system. So once these aggregates build up, they actually can feed back and attack certain parts of the cell and disable them and basically wipe out the lysosomal system, make it dysfunctional. And that allows the protein aggregates to build up even further. It's a self-propagating mechanism. And so we think if we could intervene in that mechanism, in that cycle somehow, that will cause the cell to go back to its normal state and hopefully some therapy for patients. So we're really focused on figuring that out mechanistically because we think if we do that, we will uncover many more therapeutic targets. And I think the final thing would be, I hope that in the future, there are more biomarkers available for tracking the progression of neurodegenerative diseases and determining if putative therapeutics have some effect in patients. There are a few new ones that have come out. It's not as bad as it has been in the past, but there are synuclein aggregates, for example, that can be detected in cerebral spinal fluid of patients. And there's assays that you can tell if there's synuclein aggregates in the nervous system. And so that's been a major, I think, advance that has come about recently. But I'm hoping that more blood biomarkers will be available in the future, which I think would really give us an advantage on therapy development.
the description of this kind of nefarious behavior of these proteins attacking the cell or reproducing, and you said it kind of acts like a virus. There's hypothesis out there that's been written about, is Alzheimer's a virus? Is that the connection that where people, is it just a kind of description, or do you think it could be a virus?
I don't think it's contagious like a virus. When people talk about transmitting neurodegenerative disease, I think there's really only one example of that happening and that's through prion disease. Requires a rather large exposure to the misfolded protein in order to transmit it. Nothing like that is, as far as we know, is happening in Alzheimer's or Parkinson's. What I think what we do know is that within the cell, it's acting, I think, internally like a virus would, in the sense that it can promote its own growth and within the nervous system transmit from one neuron to the next, but would not be able to get out and spread between people like a virus could. So this would be all an internal system that we think these properties are very interesting. The fact that that these protein aggregates are able to self-seed, corrupt the normal form into the misfolded form, and some of the cell biology assays that we've been doing, as I mentioned, indicate that it can disable the lysosomal system, which would give it even more opportunities to grow and take over the inside of the neuron.
Yeah, it's fascinating. It's scary. It's nefarious that this can happen. And you said your research is very kind of that early stage of just understanding that why is this happening? Do you find it difficult to get support for funding, or just because it is something that's very kind of basic that you have to understand before you're the potential for a therapeutic is, a longer term for you. How easy is it to get support, or how hard is it to get support?
It's difficult to get support. It depends on the idea and how risky it is. It's hard. It's certainly hard to get support for risky ideas that have little foundation. But if you have a solid foundation, the NIH funds research like this all the time because they understand that mechanism is the way to discover novel therapeutic targets and therapies. We have to understand the underlying disease progression and mechanisms in order to get these new medications. There are some foundations that will fund early stage, and there's others that don't. They're just interested in more mature projects that are ready to be small molecules that are ready to be put into patients, for example. So it really, really depends on the project, I think. But I think the main setback for us is that a lot of these grants are very short term and 2 to 5 years, I say, which is goes by pretty quickly. And renewing them can be a challenge sometimes. So it's difficult, but I would say not impossible to get the money from you.
What would you do with the grant that went out 7 to 10 years? Does that accelerate the understanding, or like what would be the difference if these grants were longer term?
Yeah, I think what you would have these opportunities to go deeper into mechanistically what you want to study. It definitely gives you a little more freedom. There are a lot of unanticipated problems that come up during grant projects, and not having to have to worry about, I think, the timing of it, would be a big advantage, because it's just, it's stressful, and as I said, it allows you to think, I think, more freely about what you would do in the context of, let's say, 10 years instead of 2 to 5 years. Sometimes projects have potential, but you're not getting positive results within a few years, and it's hard to get things with negative results renewed. So I think that's where it would be very helpful.
Yeah. And that doesn't mean that you wouldn't be publishing and getting information out. It would just mean that it just takes away, that worriment that you've got to produce quickly or you're going to have to stop, I guess.
Yeah, yeah. As I said, I think it's something that more of a mindset where you can go deeper and mechanistically what is occurring. And for our research, at least because we're focused on early stage disease processes, I think the longer term projects are more important.
Yeah. Well, Joe, as we kind of wrap up here, did we miss anything? I think we hit some really great high points here. Was there anything that we kind of missed that you wish we would have talked about to kind of elaborate on what you're doing and the importance that you're doing? The importance of the work. Any other thoughts there?
I think we covered a lot of it. I like this, what we are doing to study mechanism is we're trying to connect rare lysosomal diseases with common neurodegenerative diseases. And I think we touched on this in the beginning a little earlier. There are some common variants there that a lot of people don't see. It's maybe more difficult to see how the study of a rare disease that maybe affects 100 people in the whole world, how can that affect people with Parkinson's disease? And the fact that we're studying these rare variants and rare cases, it gives us this nice opportunity to look at how, because we understand what's initiating the disease in those rare cases, we know what the genetic variant is that's causing the problems. That opens up a lot of doors for us, and we can then study downstream of that problem and really accelerates how mechanisms can be discovered for neurodegeneration. That's what I think is one of the more important important things to, I think, stress from the work in our lab.
Yeah, and I think that's it is unfortunate that the really rare diseases don't get the information, don't get the kind of the visibility for funding or therapeutics or that work, but tying that through the kind of the mechanism of what is going on in those diseases in the cellular level to more kind of normal diseases, neurological diseases. It seems to me that there'll be kind of a full circle back to these rare diseases, because as you understand to the normal degenerative diseases, that there's some learning there back into the rare diseases.
Yeah, there's lots of connections, and they're just really starting. Genetics has really started to uncover these, and I think that's what's going to lead into new understandings for the disease and therapeutic targets. Like GBA1 is a good example, because now there's lots of people interested in developing targets against that will activate that particular enzyme. So before the connection was made between GBA1 and Gaucher's disease with Parkinson's, people were not really looking at that pathway. So there's lots of opportunity there.
Yeah, yeah, and so glad that you're doing this, and you're spending your time and dedicating, your passion, your time, your days to try to try to understand this, because as important as we talked about earlier. I know very few people that don't have someone in their family or know somebody that's, not affected, right?
Yeah, it's a major problem.
Yeah. Yeah. So good. So how do people get in contact with you if they want to learn more about what you're doing, or, find out what they're doing or support you in whatever way? How do people get in contact with you?
Yeah, I think the best way is probably email through my Northwestern account, which is Mazzulli, Jmazzulli at northwestern.edu. I also have a webpage on the Northwestern Neurology site that if you searched my name, you'd be able to find it pretty quickly.
Yeah. Well, thank you again. I really appreciate your time. You explained very complicated ideas and processes in a way that that we can try to understand better what's going on. And you're that front end, if you would, of really understanding what's going deep on inside our brains, our bodies. And so I really appreciate it, Joe, and thank you very much. And I love the focus. So thank you.
Thank you. Thank you for having me. It's been fun.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.